So as Dan mentioned, my lab has been developing computational methods and tools to infer functions of genes and pathways in plants. But today I'm going to tell you about a very recent work on developing an algorithm to rank candidate genes in quantitative trait loci to help speed up the discovery of causal genes underlying natural variation of quantitative traits. So natural variation in plants has had a huge impact on our society, whether it be through domestication of wild species that led to the agricultural revolution a few thousand years ago, or through breeding efforts that increased yield in wheat and rice that led to the Green Revolution a few decades ago. So a big question on many of our minds today is whether we can leverage plant natural variation for making plants a viable source of green energy. And there are a lot of interesting projects that are being carried out to address this challenge. And one of those projects is a consortium that's led by Tom Brutnell, where we're trying to map biomass and drought-tolerant related QTLs in Setaria using recombinant inbred populations. And so different labs in this consortium are mapping a variety of traits like crown root development, shoot morphology, different types of photosynthetic physiological traits, as well as transcriptome and translatome profiles, and are building a lot of QTL maps. And so we're starting to be able to build a trait by genome location map or matrix where we can identify QTLs that control several biomass and drought-tolerant related traits. Unfortunately, these QTLs are quite large in size, about 5 to 10 centimorgans that harbor many hundreds of genes. And so to find the causal gene in these regions is quite a challenge. So traditionally, to find the causal gene, fine mapping is used. Then this involves generating higher resolution mapping populations and phenotyping them, which takes a lot of time and effort. More recently, GWAS studies seem to be a viable alternative, but identifying the right set of accessions that are relevant for the trait of interest, as well as phenotyping and genotyping them, is not trivial either. And so people have also taken some educated guesses about the functions and expression patterns and pathways of the genes in a QTL region. And this has worked sometimes, but it can be quite subjective, as you can imagine, and somewhat biased and not too scalable. So we thought that perhaps we could use a machine learning approach to try to make this educated guessing process a little more systematic and objective as well as scalable. So today I'm going to be telling you about the basic approach that we're using to develop this causal gene inference pipeline using machine learning, and then how we tested the approach on Arabidopsis and rice as proof of concept. And at the end, I'll try to describe how we're thinking about extending this approach to species with very limited or no training data. Okay, so this is an illustrative example of how machine learning works. It's a very popular method in computer science that's used to classify things. And so if you want to build an algorithm that can take an image like this and say it's a cat as opposed to a dog, what you do is you provided a lot of different pictures of cats as positive training data, and a lot of pictures of dogs as negative training data, and using the characteristics or features that can distinguish between these types of pictures, you can train an algorithm to create a boundary that could maximally separate the cat pictures from the dog pictures. And so this diagram shows you a two-dimensional space, feature space, represented by x1 here and x2 here. But you can imagine that if you use a lot of different types of features, you can create a multi-dimensional boundary that can maximally separate the cat images from the dog images. So this is the basic idea that we're trying to use to develop an algorithm that can distinguish a causal gene from a non-causal gene in a QTL region. So for positive training data, we're using cloned causal genes from plants. And for negative training data, this is often a tough problem in machine learning because often you know what is a positive training, sort of what positive example, but you often don't know what's a really good negative example. So in our case, as negative training data, we're using the rest of the genome with an assumption that most genes in the genome are not likely to cause phenotypic variation in natural populations. And so we used a lot of different features that I'll describe to you a little bit later to build this model. Now, initially, this algorithm didn't work very well, and it turned out that it's because the size of the negative training data compared to the positive training data was too large. And so the algorithm was able to predict non-causal genes pretty well, but it was pretty bad at predicting the causal genes, which is kind of the opposite of what we want. And so to get around this problem, we developed an ensemble learning and ranking approach where instead of building just one model, we built hundreds of models. And for each model, we took a different subset of genes from the genome as negative training data. And this number of negative training data set was made to be the same as the number for the positive training data. And as I said, for each model, we took the negative training data from different parts of the genome. And then we had each model classify a gene to be either causal or non-causal, and then summed up all of the votes of the models to rank the genes in a QTL region to have a certain probability of being causal. So this is the basic approach that we've used. And some of you might be wondering, do we have enough positive training data? So it turns out the answer is kind of yes and no. There are about 300 causal genes that have been cloned that are responsible for natural variation in plants, and about half of them come from Arabidopsis and rice. The rest of them come from different species. And when you look at the different categories of traits, they fall into mainly 5 categories: metabolism, development and growth, flowering time, pathogen resistance, and abiotic stress. As you can see here, the number of causal genes involved in abiotic stress is much smaller than the rest of the trait types. And in fact, most of these genes actually come from Arabidopsis. So this is a pretty understudied area in quantitative genetics, especially for crop plants. So to build a supervised learning algorithm like this, you need at least 50 positive examples to be used for both training and testing. So we tried to develop the algorithm initially for Arabidopsis and rice. So what are the features we used? So we took advantage of the sequenced genomes and, of course, all of the different types of annotation tools that we and others have developed. So the features can be sort of grouped into 3 categories. So first, we used different ways of inferring molecular functions of genes. And so different gene classes were used, as well as different metabolic enzyme classes. Another main category of features we used have to do with polymorphism types, both the positions of the polymorphism within the gene, as well as the functional consequences of the polymorphism, such as, does the polymorphism lead to a stop codon or a non-synonymous substitution? And if it's a non-synonymous substitution, does it lie in a conserved residue? The features that are marked in the gray boxes here are features that have to do with functional features on the DNA, such as microRNA binding sites and transcription factor sites. And these gray boxed features are things that we're in the process of incorporating, but have not been added to the model that will be described to you. Finally, we've used these genome-wide co-function network links that we built for Arabidopsis a few years ago, and that In-Suk Lee at Yonsei University built for rice subsequently. And these networks are based on functional linkages between genes that use evidences from a variety of data, such as co-expression, physical interaction, genetic interaction, and whatnot. So using these features, this is the setup that we had for Arabidopsis to prove the concept. So we started out with about 50 curated causal genes and set aside about 80% of those genes for positive training, and then took random genes from the genome as negative training. And using the features I just described to you, we trained an algorithm to build a predictive model. And then we used the 10 causal genes that we set aside from training. So these are genes that hadn't been a part of the training to test. And for each of these causal genes, we took 200 neighboring genes as negative genes. And so we labeled and ranked all of these genes to see how predictive the algorithm was. So an ideal performance would look something like this. So for all of the test genes that you try to predict, we would want to see the causal gene to be ranked on the top of the 200 genes. In reality, on Arabidopsis, this is the performance we observed. So it wasn't so bad, but I think it's not spectacular. It's not the ideal case, but I think there's room for optimization and improvement, for example, like adding additional features and so on. So what we found was that 90% of the time, we were able to put the causal gene within the top 20%. And about half of the time, we were able to put the causal gene in the top 10%. So, so we were able to narrow down the candidate genes from about 200 to 20 to 40. And as a start, I think this is not so bad. It gives us hope that we could sort of improve this. And so then we also asked whether this would work for rice. And so we built a rice model and tested it on rice causal genes. And we saw exactly the same performance. So what are the features then that are predictive in these two models? This was kind of a surprise to us. For the Arabidopsis model, by far, the number of edges in ARANET, this co-function association network, was the most predictive feature. And so this is basically the number of associations the gene has to other genes. So these are the hub genes, basically, that seem to be more likely to be causal. The second best feature was non-synonymous substitutions in conserved residues. And so this was also kind of a surprise to us initially because we thought more drastic polymorphisms like those that give you stop codons we thought would be more causal than non-synonymous substitutions. But when we thought about it a little more, it kind of made sense because these are polymorphisms that have been that are responsible for phenotypic variation in natural populations. And so more drastic mutations such as gaining stop codons might actually not be selected for. The rest of the features were sort of equivalent in their ability to predict, and they ranged between different types of functions like transporters and receptors and different types of polymorphisms, like insertions and deletions in up and downstream of the genes that may indicate changes in expression patterns. So what about rice? Is it similar or different? Again, we were kind of surprised that the top two features that contributed to the prediction accuracy were exactly the same as for Arabidopsis. So it was the number of edges in the rice net as the top feature, and then the non-synonymous substitutions in conserved size as the second feature. And the rest of the features that contributed to the prediction were similar to Arabidopsis, but the relative orders were kind of different. So this sort of gave us hope that perhaps we could use these models on species that don't have training data. Okay, so we tested this idea by looking at rice causal genes on the Arabidopsis model. And to our dismay, the performance actually wasn't that great. So only about 10% of the time, the causal gene was ranked on the top 10%, and only about 20% of the time, the causal gene was ranked on top 20%. And this was the same case for testing Arabidopsis genes on the rice model. So what could be the reason for the lack of the ability of these models to work across species? So we could think of a couple of reasons. I'm sure there are many more. But one main reason is that there's different evolutionary distance and polymorphism rates between the ecotypes that we use for these species. For example, the synonymous substitution rate for rice is about 10-fold higher than that for Arabidopsis. Another reason might be that the causal genes that are identified in these two species are pretty different. So for Arabidopsis, abiotic stress and flowering time genes are enriched, whereas for rice, it's mostly development and growth genes that have to do with grain qualities. And so are there ways to actually address these issues and actually build a model that's species independent? So there are a couple of ways that we are trying to go about addressing this issue. One is that we can try to normalize these features, especially the polymorphism features between species, such that we can actually scale the polymorphism rates, like synonymous and non-synonymous substitution rates, to a reference scale. And this may allow us to make a genome-independent model. And if we can do that, we might be actually able to build a separate model for different trait types because we actually have enough causal genes for different types of traits if we can combine all the causal genes from different plants. Another approach that we can take is to use an unsupervised learning where the algorithm doesn't require positive training data, And we can do this if we know which features would be predictive. So from the work on Arabidopsis and rice, we know which features actually work well for both species, and we can start to test more species like science of selection and other types of features to feed into an unsupervised learning setup. So in summary, I've told you that we built a machine learning pipeline to infer causal genes in QTL regions, and that the supervised learning approach works well for each species, but not so great for between species. And so to try to build models for species that have very limited training data, we're going to try a couple of approaches like unsupervised learning and genome-independent model by normalizing the features. Finally, I'd like to thank the people who did the work. So most of the work that I told you about today is done by Joy Fan, a postdoc in the lab, and Taehyung Kim and Jim Guo contributed a lot of the annotations that Joy used. And as I mentioned, this is a collaborative effort among several laboratories in the US and a consortium that's been funded by DOE. Thank you.
Thanks, Sue. Question, Asaf.
So I think you kind of artificially restrict non-negative examples, because rightfully you should include all negative examples, and then your accuracy drops. And generally, I think it should not work based on these characteristics, prediction of causal genes.
Can you explain what you mean by artificially restricting the negative?
Because so number of genes in genome, it's 20 to 30,000.
That's right.
And you have just 50 genes training set.
That's right.
And you say, I say 200 negative genes. And then you have
yes, okay.
high, false positive rate is lower.
Right. Actually, so you're talking about the 200 non-causal genes we're using to test, right? So we have actually tried 500 and 2,000. And in fact, it doesn't matter what the number of non-causal genes is, because actually you're trying to use characteristics that overall separate the cause from non-causal. So the probability of being non-causal and causal is the same.
No, non-causal is cloud, and causal maybe non-causal maybe everywhere. It's
well, maybe we can discuss this afterwards at the reception. The break.
Tom?
So I realize that you're limited by the number of QTL studies that have happened, but have you tried to group them into bins that might be more similar? So say a biochemical phenotype like cell wall composition in Arabidopsis versus rice, and try to make the sort of the phenotypic screens more equitable.
Right. So we're going to try to do that for Arabidopsis. I think we may be able to subpartition the training data to be able to look at specific traits and different sort of processes. And a lot of those features that kind of ranked lower in their prediction accuracy but were equivalent, I think most of them have to do with the trait types. So we can really test that idea by separating the training data for Arabidopsis. Yeah, we're going to do that.
One question here and then in the back.
So thinking about the Arabidopsis to rice comparison, I guess it's not that surprising that sort of causal polymorphisms might be very different between the species. But I would assume hub genes tend to be hub genes in both species. Is that true, or have you tested that?
That's a really good question, and we're going to sort of investigate that some more and look for additional features in these co-function networks that might be predictive. They tend to be regulatory genes that are hubs, but whether these hub genes between rice and Arabidopsis are exactly the same or how similar they are, we haven't really looked at, but we should.
Steve?
Yeah, hi. I'm just wondering if there's a way of testing not using real data but simulated data for GWAS. Have you considered this?
That's a really interesting idea. We haven't considered it, but it's certainly doable. And maybe we should, actually. That's a really good idea. Yeah, we can simulate different kinds of mutations, for example, and yeah, maybe we should try that.
Last question in the back.
What machine learning algorithm did you use for this?
Right, that's a good question. So I didn't go into the details of the algorithm. So the one we use is random forest, which is an ensemble method that uses decision trees. But we also tested support vector machines, Bayesian model. What was the other thing we tried? We tried about 4 or 5 different approaches, and decision trees was best in terms of recall, which is what we want in this case, because we want to have that causal gene found, whether it's like top 10%, 20%, because we can always like try to increase the precision later.
One last question.
When you look at the distribution of genes, if you compare this to, say, text mining, normally if you look at text mining and you look at the frequency of words, you'd see a zip distribution of the data. And do you see the same thing with the genes, where you actually, if you go on a gene-by-gene basis, where you see a distribution that
Right.
follows a zip distribution, or is it more, is it a flatter distribution?
So are you talking about different functional distributions of the genes? Yeah,
functional
right.
distribution.
So
If you're looking at words, and can you actually put in stop words and do many of the other things that you normally do in text mining?
put in
Stop words. So you can actually improve your training set.
I'm not sure if I know exactly what you're talking about, but we have looked at the distributions of functions, basically, in the causal genes, for example. And they are enriched in certain categories, like transcription factors, transporters, and receptors. And we thought initially this would be a really good predictive feature. But it turned out that they weren't as predictive as we expected. So transcription factor, for example, was still a predictive feature, but it ranked pretty low. So some of the a priori sort of things that we had in mind based on the differential enrichments of things in the causal genes weren't really, like there wasn't a 100% match. So I think that it's part of it is really like integrating a lot of these features. And this is where I think the subjective and the objective thing comes from. The machine learning is framework is actually able to incorporate all of the things with a sort of blind mind, whereas humans, like, if we see something that's enriched in these things, we think it must be important. So I'm not sure if this directly answers your question, but we have looked at different types of distributions.
Okay, let's thank all our speakers for this morning.